disaccharides, what did we say in the introduction? Remember, we said that we call them disaccharides because they are made up of 2 monosaccharides linked. So it's like you have one monosaccharide and another monosaccharide, they come together, and that final structure they are making is what we are calling a disaccharide. And for disaccharides, we have 3 examples of disaccharides. Those are the ones we shall focus on for today. Number 1, there is a disaccharide that you people are very familiar with. And the name of this disaccharide is maltose. The other one that you are familiar with is lactose. Then the last one is sucrose. Some of the sugars you are seeing here, the disaccharides, we even use them at home. Maybe we just didn't know. I've heard a lot of people making mistakes, calling the sugar that we take at home glucose. That is a big mistake. For a biochemist, that sugar we take at home, the table sugar, is actually sucrose. So sucrose is simply table sugar. Lactose, milk sugar. This is what is found in milk. You know that mammals, like humans, tend to provide energy to the young ones, the babies, through milk. And what is in that milk is simply lactose. Later we shall know what monosaccharides are used to make lactose, what monosaccharides are used to make sucrose. Maltose is the sugar that is used to make infant formula. Most of the infant formulas that we use, you know, there are situations where a mother cannot breastfeed a baby, maybe due to various reasons, and we tend to rush to infant formula. There are so many brands of infant formula. So those are, we normally make them from maltose. Beer, flavor, the flavors that are used when baking, they are made from maltose. In plant biology, we learn about seed germination. When a seed is germinating, it also uses maltose as a source of energy. I don't want you to forget the most important thing that you should remember about disaccharides. It's not just their names. You should ask yourselves one important question. What monosaccharides are used to make maltose? What monosaccharides are used to make lactose? What do we use to make sucrose? Maltose, we use alpha glucose. We combine it with another alpha glucose. How do they combine? Carbon number 1 of the first sugar combines with carbon number 4 of the next sugar. And we have made maltose. Lactose, we get beta glucose. We combine it with the beta galactose. Again, carbon number 1 reacts with carbon number 4. What about sucrose? We use alpha glucose. We combine it with beta fructose. But here, we don't use carbon number 1 and 4. We use carbon number 1 and number 2. So it's like you get one glucose and another one, you combine them, and when you combine them, they become one structure. That one structure is maltose. And the bond between them is called a glycosidic bond. So a glycosidic bond is a bond that links successive monosaccharides to make huge carbohydrates. So we are calling it glycosidic bond. If I was to ask you a question to state the type of bonds or the type of bond that is formed when maltose is made, If you give me this answer, I might not give you full marks. Why? Because the name is not complete. When you are stating the name of the bond, me reading that name, I should be able to know or to see the type of sugars that were used, in this case alpha, and the carbon atoms that were involved in the reaction, in this case carbon number 1 and number 4. So the complete name of this bond between them is not just glycosidic bond, it's alpha, because we used alpha glucose, 1,4 glycosidic bond. That is the complete name. Alpha 1,4 glycosidic bond. Why? Because we used alpha glucose molecules and we reacted carbon number 1 with number 4. Same applies here. Are we using alpha? No. This time around we are using beta. So it's beta 1,4 glycosidic bond. What about the last one, sucrose? Glucose is alpha, fructose is beta. And we are using number 1 and number 2. So the complete name is alpha beta, because we are using alpha and beta, 1,2 glycosidic bond. When you go to the notes that you've been given, you see that there are diagrams there demonstrating how sucrose is made, demonstrating how lactose is made, demonstrating how maltose is made. For you to capture the concepts from this lecture, I'm going to pick one, maltose, and I'll demonstrate with maltose. The process that will happen here is similar to the processes happening in the other disaccharides. So let me demonstrate for maltose. What are we saying about maltose? Maltose is made up of alpha glucose and another alpha. How do you demonstrate? Simply draw 2 alpha glucose molecules. Remember what we said, carbon number 6. Remember, we are saying alpha. If it is alpha, where is the name alpha coming from? From the fact that on carbon number 1, the first carbon, the first generic carbon, what OH is below and H is down, H is up. That is where the name alpha is coming from. We are calling it alpha for that reason. But we know the rest, how they are. There is OH, H, H, there, OH, H, and then here CH2OH. That is the complete structure of glucose, alpha. Let me draw another one next to this one. Carbon number 6, CH2OH. Again, same structure. We are calling it alpha because of number 1. Because on number 1, OH is down, H is up. But what is there? Already we have OH, OH, and here OH. So what I've done is, I have drawn one alpha glucose, and next to this one is another alpha glucose. That is what is here, alpha, alpha. Carbon number 1 of the first glucose will react with number 4 of the next. You can see number 1 is there, and number 4 is there of the next. The first one would donate OH and gets H from the next one. Then in the end, they create water molecule. So when carbohydrates, when monosaccharides are coming together, when carbohydrates are being condensed, they liberate water. Water is given out. How is it created? The first one donates OH here and gets H from the next one through the OH there. And when that happens, the final structure will look like this. You can complete the other parts, but I'm interested in showing you the structure. You can see that when you remove the OH and H, you remained with that oxygen. That is the oxygen I'm showing you there. So it acts like as a link between this sugar and this sugar. But what they are making is one structure. And the name of the bond between them, because we are reacting carbon number 1 and number 4, and this is alpha, this is alpha, this is the one we are calling alpha 1,4 glycosidic bond. I want you to go and practice this reaction using lactose and sucrose. Use the same method. For lactose, go and draw beta glucose. Next to this, that one, another beta galactose. Then you link them. Also for sucrose, you should draw alpha glucose and beta fructose. Link them so that you practice on how the glycosidic bond is formed. Now that we understand formation of this glycosidic bond, it's very easy for us to proceed to go to other huge carbohydrates like oligosaccharides and poly. Why is it easy? The same process that has happened here, where we are linking one sugar to another, it continues happening. Another sugar comes there, another one, another one, another one. As they bond to each other like that, they in turn make very big structures, huge structures. That's where oligosaccharides and polysaccharides fall. We are not going to spend time on oligosaccharides. There is not much to say about oligosaccharides, but there is a statement I want to give to you. And the statement about oligosaccharides is that in the introduction, I told you that the third function of carbohydrates is that they are used to make cell surface receptors, which are used in cellular communication. I should state to you right now to say it's not actually whole carbohydrates that are suitable for that process. Making of receptors works very well with oligosaccharides. Even synthesis of hormones and other important substances, they work well with oligosaccharides. Having said that, I'm going to now finish the discussion of carbohydrates by looking at the last group of carbohydrates, which is called polysaccharides. So we are looking at polysaccharides as our last discussion. What did we say in the introduction about polysaccharides? We said that polysaccharides is simply more than 10 sugars. If I was to use cartoon drawings, it simply means one monosaccharide links to another one, another one, another one, another one, until there are more than 10. So what is the name of the bond between them? Simple. We called it a glycosidic bond. To be specific, the 1 and 4, so 1,4 glycosidic bond. That's what it means by polysaccharides. So our discussion of polysaccharides, I want you to understand as we discuss this important concepts about polysaccharides. What are the important concepts on polysaccharides? Number 1, sometimes when we make them, we can use the same monosaccharides. What does it mean by same monosaccharides? It simply means maybe glucose, 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 or maybe galactose, 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 or maybe fructose, fructose. What it simply means is that sometimes when we are making a long chain of monosaccharides to make polysaccharides, we use same sugars. Sometimes when we are making use same sugars, the name of the polysaccharide is homopolysaccharide. So homopolysaccharide simply means you are using same sugars across. However, sometimes we might use different sugars. We mix them, not one type. Maybe you have glucose. After that, galactose. Maybe again glucose. The fact that you have more than one type of sugar linked to it in this structure, we can't call it homo. We call it hetero polysaccharides. Two concepts. When you are making polysaccharides, you use similar sugars throughout, homopolysaccharides. When you are making polysaccharides, you use different sugars, you mix them. That is heteropolysaccharides. Concept number 3. In the previous demonstration, I told you that actually it's like one sugar, another sugar, maybe another one. They continue linking to each other. And the linkage I showed you is this one. And the name of the bond, if you are remembering, is 1,4. 1,4. Because you are using carbon number 1 and 4. If we were to complete the structure on number 6, here there is CH2OH, CH2OH, CH2OH. What concept do I want to demonstrate to you right now? If this process is to continue and you have more than 10 sugars, then the structure you've made is a polysaccharide. Now, for polysaccharides, there is something very important that happens. After a straight chain is made like this, some polysaccharides, instead of a sugar coming here to bond to this one, the sugar comes on top of another sugar. Like that. And when it comes on top of another sugar, instead of the bond being number 1 of one sugar reacting with number 4 like this in a straight chain, it will be number 1 of the sugar on top reacting with number 6. The same thing happens. Donates OH, gets H. Water is liberated. But the bond is not 1,4. It's 1,6. If I'm to use cartoon drawings like this, they will look like this. It's like you started making a big chain. You make a long chain like this. Then after that, you start forming a branch. So some carbohydrates, specifically polysaccharides, when we are making them, they make a branch. And branching happens at number 1 of this sugar and number 6. While a straight chain is between number 1 and number 4. That is the third concept on carbohydrates. Having discussed the 3 concepts, number 1, making polysaccharides using the same monosaccharide. Number 2, making polysaccharides using different or a mixture of monosaccharides. Number 3, branching polysaccharides. Let us look at the 4 groups or examples of polysaccharides as we summarize the study of carbohydrates. 4. What are the different examples of polysaccharides? Number 1, there is a group of polysaccharides that we use for storage, so we call them storage polysaccharides. What it means is that when we make them, we just make them for the purpose of being used as a storage form of carbohydrates. For future use, for example. The second group that we shall talk about is structural polysaccharide. Structural. As the name suggests, storage for the purpose of being stored. Structural, we make some polysaccharides for the purpose of them being used to make important structures in both plants and animals. Let me give you examples. In plants, Plants, they tend to store excess glucose in their seeds as starch. So starch is an example of a storage polysaccharide. Number 2, animals. We also tend to store excess glucose. So when we have too much glucose, we store excess in the liver and the muscle as glycogen. So glycogen is an example of a storage polysaccharide. What about structural? Plants, sometimes they synthesize a polysaccharide called cellulose, which is used to make the cell wall of their cells. It's an example of a structural polysaccharide. Animals, they synthesize what is called chitin, which is used to make the exoskeleton of insects. Those insects you see, beetles with a very hard cover, that cover you see on beetles is simply chitin. To summarize my discussion for today, I want you to understand what types of sugars do we use or monosaccharides to make the examples on the board. So to make glycogen and starch, we use the same sugar. It's alpha glucose. To make cellulose and chitin, we use the same sugar, beta glucose. But how are they connected? For the purpose of this lecture, I'll use circles to demonstrate a sugar. But in your mind, when you see me doing this, you should know that it's the full structure I'm talking about, the structure of a sugar. So a circle in the discussion of polysaccharides represents the full structure of a sugar. So starch. Starch is of two types. There is a starch that is called amylose, and there is a starch that is called amylopectin. What are the differences? Amylose is not branched. Remember, what does it mean not branched? It means that alpha glucose, alpha glucose, alpha glucose, alpha glucose, just a straight chain. When we come to amylopectin, it is branched. What does it mean by it is branched? After making a straight chain, you come and make a branch somewhere like that. If you can even make it there. So amylose only has alpha 1,4 glycosidic bond. But amylopectin is going to have 1,4 in a straight chain, and where it is branching there, it will have 1,6. So the two types of bonds will exist in amylopectin. Alpha 1,4, alpha 1,6. So remember, if a question is asked, what are the differences between amylose and amylopectin? Amylose is not branched and has only 1,4, alpha 1,4 glycosidic bond. Amylopectin is branched and consists of both alpha 1,4 and alpha 1,6. The same way you are seeing amylopectin is similar to the structure of glycogen. Glycogen is also branched, just like amylopectin. And it has both alpha 1,4 and alpha 1,6, just like amylopectin. But it branches systematically, meaning it doesn't branch anyhow. It branches after 8 to 10 sugars, meaning 1, 2, 3, 4, 5, 6, 7, 8. That's when it will make a branch. Again, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, branch. But this one, it doesn't branch systematically. And this guy is more branched. Glycogen is more branched than amylopectin. But they are similar because they both have 1,4 and 1,6 glycosidic bond. The last two examples of structural polysaccharides, cellulose and chitin, they actually look like amylose, but uses beta glucose. What does this mean? If you were to draw the structure of cellulose, it's just like amylose. What does it mean? Not branched. And we have only beta 1,4, not alpha, because we are saying that for the two, we use beta glucose molecules. So if you are to draw using the circles I'm using, the structure of cellulose will be a beta glucose linking to another beta, linking to another beta, linking to another beta. The process continues. That is the structure of cellulose. How does it differ from chitin? For chitin, it is similar to cellulose. It is also made up of beta glucose molecules, and the type of bond present there is beta 1,4. But the difference is this one. After you link beta glucose molecules together, for chitin, at the end, we conjugate a protein. We attach a protein to this structure. And the moment we attach a protein, the name of the structure is chitin. So that hard structure you see, the hard cover on the back of beetles, that is made up of chitin. It's just a composition of some proteins and carbohydrates coming together. We have come to the end of the topic on carbohydrates. Before I go, I should mention to you that ensure that, number one, you understand the functions of carbohydrates. After understanding the functions, ensure that you know the classification, the different groups of carbohydrates, monosaccharides, disaccharides, oligosaccharides, and polysaccharides. And in each of the groups, you should be able to give examples. Where necessary, know their structures, because that's where questions will come from. Thank you very much for being with me. Next time we come, the topic we shall go to is another important biomolecule. So our next discussion will be on lipids. We shall try to understand what are the functions of lipids, what is the classification of lipids, what is the structure of lipids, the same way we have discussed carbohydrates. Thank you so much.